So my task today in the 22 minutes is to try to make a connection between what we're trying to do. There we go. What we're trying to do, I think, in a number of different disciplines, which is to try to understand the underlying biology of this disorder that we call the autisms or autism spectrum disorders. And I think Peter presented the level of complexity that we're trying to deal with. And if we were thinking about this as a model system to do experiments, and we would probably throw our hands up and say, let's move on to the next model system, because it's so complicated. And I think many of us who come from outside the field have focused almost entirely up until recently on trying to understand the disorder from the diagnostic criteria that are used clinically. And I think what Peter said is very, very important for those of you who don't study this disorder, is that this is a very complicated set of behaviors that are exhibited in a very heterogeneous manner across the population. And that means that the neurobiology, the underlying biology, is also extraordinarily complicated. And it's not going to be a simple set of answers. And we have to move beyond the diagnostic criteria way of thinking about this disorder, the way sometimes we think about diabetes, high blood sugar, cancer, cells are dividing, and move to really what we should be more comfortable with as scientists, which is to think about the biology and the behavioral components that we can investigate with far more precision. So I put my acknowledgement slide in the beginning, and most of the work I'm going to talk about today is work that Dan Campbell, who's a research assistant professor now here at Vanderbilt, has done. There are other people who are highlighted here, and some of the collaborators on the genetic studies, including Jim Sutcliffe, Tony Persico, collaborator in Rome. And I gave a talk to students at Columbia last week and said, pick your collaborators wisely. It's not necessarily how good they are as scientists, but it's where they're located, and Tony's in Rome. And so that's not a bad place to visit. And this is some of the resourcing for the work that I'm going to be talking about, including Mari, of course, and some other resources. So this is a diagram that Dan Geschwin and I put together when we were on the phone with each other. And it ended up being a bit of a nightmare, I think, in some ways, because it's very complicated and it's hard to see on this screen. It's even hard to see when you read the paper itself. Because what it's supposed to convey is that the etiology, the causes, the underlying way that we're thinking about genetics, really, we don't need to do another single experiment to know that there are going to be different combinations of genes or different genetic causes that link to the environmental components that may be causes or modifiers of the disorder. And we know that because there are already a lot of syndromes that have co-diagnosed autism spectrum disorder at a fairly high rate, 50% or more. And we know specifically what gene causes that syndrome, and that's shown on the bottom. And therefore, we know that there must be different genetic causes. On top, we tried to capture, what we're trying to capture is thinking about the disorder more broadly than the DSM-4 criteria of social relatedness, communication, and focus on restricted interest, to think about some of the things that have been reproduced over and over again by different laboratories that get us to think a little bit more broadly about the biology. One of the things that we know neurobiologically, we don't have an explanation for it, but it's been reproduced by the Corshane group, by the Dawson group, by the Piven group, is that if you look at the population of individuals with autism spectrum disorder, there's a difference in the rate of growth of their brains and their heads. And there's some debate about whether this onset is at 6 months or at 9 months or at 12 months. But if you look at the population, it looks like, and that's shown in the red line, that group starts to show accelerated growth of the brain somewhere in that first year of life, somewhere between 9 and 12 months, probably most people will agree with, and that there's a fraction of those children that end up being macrocephalic, meaning that they're in the 95th percentile or greater in terms of head size. And what that tells us, I think, what we can conclude is that there's a different trajectory of development for those individuals, and probably for many of the individuals who are diagnosed with autism spectrum disorder. We don't know what the underlying neurobiology is, but we know that that trajectory is different. And if there's anything that we've learned over the last 10 or 15 years about development, is that how you get there is very, very important. So that difference in curve is telling us something about altered biology, and it's telling us something about altered biology early on. The other thing we know from studies of Marcel Just and Nancy Minshew and others, Peter mentioned a bit about some of the ERP work that he and others have done, is that we think about this disorder, and Dan Geschwin and I highlighted as a syndrome of, that you could classify as disconnection, meaning that the connectivity between different brain areas is not functioning properly. We think about that as structural disconnectivity, but in fact it could be molecules that are causing the circuits not to function as they normally should. And Marcel Just and Nancy Minshew did an experiment using functional magnetic resonance imaging to essentially show what's shown on that graph. The top is the control group, the bottom is the group of individuals with autism spectrum disorder, is that different brain areas that should be activated coincidentally when an individual is performing a task are not being activated coincidentally. And that tells you that there's some problem with the connectivity, some problem with the information flow between brain areas. That's a biological prediction from this, and what we're really struggling with is then trying to understand what specifically about the developmental neurobiology of the brain would generate that kind of a picture in terms of information flow not being appropriate. The other thing that we know that's been replicated in a number of different studies from a biological perspective is that there are different parts of the brain that contribute to the difference in head size and in brain size. This is work from Martha Herbert, who did this work and published this paper in 2004 from the Massachusetts General Hospital group, in which she essentially showed that there were selective parts of white matter that carries the fibers from one brain area to another. This is white matter, but she's also shown this with gray matter, that show increases in size in the population that she looked at. And that fits with what we know from the head circumference data, that there is an accelerated growth, and then there's a flattening out, so that there's an accelerated growth early, and then what we would expect from typical development with continued increase in size doesn't happen generally in this group. So they mature earlier in terms of size of the white matter and gray matter, and then there's a flattening. And that's another neurobiological indication that there's a problem initially in some of the features of development that occur during this period of time. What are those features? Synapse formation, myelination, increases in blood vessels. Those are the kinds of things that we think may be contributing to this atypical brain growth. Now, I already talked to you, and Jim Sutcliffe already mentioned that we know that there are a number of different genetic syndromes where we can identify the gene, and they're listed here, that have high incidence of co-occurring autism spectrum disorder. And I know that there's debate about whether this is real autism, whether this is kind of autism in some of these syndromic disorders, but I think Peter mentioned Fragile X. We know that all these others with different mutations of genes generate a very high prevalence of the disorder. And what do they have in common? Well, one thing they have in common is a disruption of connectivity. The disruption of connectivity caused by different genes, not so surprising, because if you go to a current database, there are right now approximately 14,000 genes that are listed as being part of the synapse. That's the connection between nerve cells. 14,000 proteins encoded by different genes are at the synapse. So if you play the 6 degrees of Kevin Bacon, you actually don't have to do more than 1 or 2 steps to link whatever protein you're interested in the developing synapse, because about 2/3 of the genome has already been identified as being there. That's a big problem in trying to understand the underlying neurobiology. If 2/3 of what you're working with is at the place that you think is part of the problem, of course everything is going to be related to everything else, just like Kevin Bacon's related to every actor who's ever worked in Hollywood. Okay. So I took this slide from, who did I take this slide from? I can't remember. Who did I take this slide from? You don't know? Okay. From you. Did I take this from you? No, I didn't take this from you. Francis Collins, who's head of the Human Genome Project. And we're right now in this kind of dimension of doing genome analysis, define association of risk genes, and defining disorder-related variations in sequences of risk genes, what Jim Sutcliffe talked about today. Here's the real challenge. My charge is to bring it back to the brain science. We need to determine the biological implications of those variations. It's one thing to identify a genetic marker. It's another thing to figure out what that means biologically. How does that genetic marker change the underlying development of the individual and the brain? We need to determine influence of variations on the phenotypes of the disorder, some of the phenotypes that Peter talked about. Do those variations, together with environment, which we're going to hear about a lot today, have an influence or an impact on the specific features beyond the diagnostic criteria of autism spectrum disorders? And then finally, can we use this genetic information to evaluate the usefulness for diagnosis or for figuring out better ways of designing treatments, or even predicting which child will respond to which treatment, something that Paul Yoder and I have talked about quite a bit. Now, if you notice, what do you notice about this slide? Take off your glasses. I can't see I can see the top. Yeah, I can see the top. So we can do genome association now, but we're not even close to getting to the bottom. And the goal, I think, for all of us in the field is to increase the font size on the bottom, because that's ultimately what we're trying to do. Okay, so I'm going to talk to you. How much time do I have left? Scads? I have scads of time left, according to Beth Ann. Scads is like sugar, by the way, Dan. I'm going to talk to you today to give you an example of how we're proceeding. This is just one example. There are many people in the field who are, I think, taking great approaches to trying to understand the biology behind or underlying the autisms. This is, I got 15 minutes, perfect. This is images that were generated by Micah Bergman and Matt Judson in the laboratory, localizing a gene that we focused on because it has an interesting expression pattern in the cortex and in the amygdala and in the lateral septum, areas that we know that are involved in behavior, particularly social behavior, in the animal. And on the bottom left, you can see that's a Western blot, that's an immunoblot localizing this protein across time in development. Remember, I'm a developmental neurobiologist, so changes over time are very important to me. What does the system look like here, and then what does it look like here? And this particular protein is expressed during the period of time in a mouse, when there's lots of really important things going on, like synapse formation and myelination and vascular development. That's the period of time. And what is it? It is the receptor tyrosine kinase MET, which was first discovered as a cancer gene. Like many genes that have been implicated in neurodevelopmental disorders, by the way, they've been implicated in cancer as well. It's just that the dysregulation of those genes in neurodevelopmental disorders may be in one direction, and in cancer they may be in the other direction. Some of those genes were already listed up there, like MECP2, UBE3A, even the calcium channels. A lot of these have been implicated in multiple diseases, but the regulation of those genes may be different, or likely to be different, from disease to disease. Now, the MET receptor, it's a membrane receptor, binds to a growth factor called hepatocyte growth factor, which activates it and runs through a signaling pathway known as the Pi3 Kinase Pathway. And the reason I put this up here is because if you look carefully, and those of you who are in the field, there are a number of genes that have already been implicated from the rare or private mutations that Jim Sutcliffe talked about. AKT, P10, NF1 is neurofibromatosis. That's a disorder that's 100 to 150 times more prevalent in individuals with autism spectrum disorder. And we'll talk about some others. But this signaling pathway, beyond the work that we've done in the laboratory, seems to be at least one point of convergence in terms of altered cellular function that we're going to hear about later this afternoon. It has interesting interactors that I'm not going to go into, and it's also MET and its growth factor are located at the synapse. It generally is not included in a diagram. In fact, what you see here is something that Dan and I put into a diagram that was published by Tony Persico, my collaborator, who seemed to forget about this gene at the synapse, in 2006. But it is at the synapse, and we know from a number of studies not from our laboratory, but from several other laboratories, that this gene, its protein, is involved in dendritic development, which is important to receive synapses, so it's involved in connectivity. It's involved in a cognitive or cellular process that underlies memory and learning called long-term potentiation. You can enhance the activity of cells to take in more information by activating this system. It's also involved in myelination, and it's involved in a number of other cellular processes during that period of time that we know that atypical brain growth is occurring. This, we believe, is one of probably a number of cellular pathways that are disrupted in autism spectrum disorder. So we set out to look at this gene and define disorder-related variations in the sequence, and this is the bottom line. I'll tell you now. There's Dr. Campbell right, I see I pointed to my slide. There's Dr. Campbell right there over on the right-hand side. I think you're wearing the same, no, you're not wearing the same shirt today, are you? Okay. Okay, you did your laundry. Very good. Dan has been studying the genetics of the MET gene and other genes, and in fact, there are now 3 genes in this pathway that have common variations that we know are showing association with autism spectrum disorder. When you have one, it's an accident. 2 is a coincidence. This is what I tell the students. 1, if the experiment works once, it's an accident. If it works twice, it's a coincidence. If it's 3 times, maybe, right? And so now there are 3 genes in this pathway that have been implicated from the work that Dan has done, where the check marks are MET, SERPINE1, and PLAR. I'm not going to talk about those today. I'll talk mostly about MET. And we actually know some of the transcription factors, some of the proteins that are shown here, LHX2, PC4, or sub1, SP1, that are disrupted by environmental agents, and that we know can alter expression of this gene and other genes as well. So Dan has done a number of studies, very large numbers of families, to look and demonstrate that there's a certain marker, a certain allele, one particular base change that shows an overtransmission from parent to child in autism spectrum disorders with a very high p-value or significance value. And when we look at cases and controls, just simply looking at a typical population without the diagnosis compared to those who have the diagnosis, so you can analyze these in 2 ways. You can analyze transmission from parent to child, or what's the distribution of this allele or this variant. It's in about 47% of the controls and about 58% or something like that of individuals with autism spectrum disorder. So it's common allele. Many of us in this room have this allele, what we call the C allele. It doesn't mean we all have autism. This is not diagnostic for autism. It increases risk, and the risk ratio is about 2.25 or so, which is modest. We also know that this gene may be involved in copy number variation. In a paper that was just published by Steve Scher in 2008, 2 of the 26 de novo or non-inherited copy number variations fell right on chromosome 7, right in this gene, and disrupted this gene. So like Jim talked about, certain genes may be implicated in disorder. Some of them may be involved with common variations that increase risk and interactions with environmental agents. Some of the implications for these genes may be due to de novo or new or private mutations that just happen in the child that were not passed on from mom or dad. So what are the influences of this variant on the dimensions of the disorder? This is getting to what Peter talked about. Basically stratifying individuals based on some common elements that we feel comfortable with and see whether this variant is enriched in that population, essentially, rather than looking at the entire spectrum, which is very heterogeneous. So the one that really hit home to us was when we took our data from the Autism Genetics Research Exchange, DNA bank, and DNA that Jim Sutcliffe had from the Tufts and the Vanderbilt collection, and Tony Persico from Italy, and basically we stratified it between multiplex families that is more than one child with autism, and simplex families, only one child. And while we admit that there are difficulties in defining a simplex as a simplex because there may be the first child with autism and then you stop, don't have children again, there is a clear segregation. That is, this variant is enriched in the individuals in the families with more than one child who has autism. And this is not the first time that there's been a suggested difference between multiplex families or families with more than one child versus one child only. This study that came out of Joe Piven's group highlights the fact that if you look at the broader autism phenotype in parents, you can see that in the MIAF, those are the multiplex families, that there's an increase in the broader autism phenotype expressed by parents compared to simplex families. So from a behavioral or phenotype perspective, as well as a genetic perspective, we may be sort of in this gross level being able to separate out the multiplex and the simplex groups and analyze then what the underlying biology might be in those groups, which may be, in fact, very different, keeping in mind that there are 14,000 proteins that we're dealing with, and we believe it's connectivity. The other way that we've stratified is by looking at co-expressing medical conditions, which we believe may be a reflection of the fact that this gene that we're working on, as well as other genes, play roles in different organ systems. It's no longer the case that we think about one gene, one function, or one gene, one organ, like the brain only, but these genes operate all over the body during development. So we focused on gastrointestinal function, and Dan has a poster this afternoon that will highlight this. And the reason is that there's a lot of reports in the literature, although controversial for reasons that I'm not going to go into now, that there's a high co-occurrence of gastrointestinal disorder in children with autism spectrum disorder. Most of them are positive, there's one that's negative, and there's some debate about thinking about trying to link the gastrointestinal disorder to causing the brain disorder. And we've looked at it in a very different way. This gene is involved in parallel developmental processes. Those are what the arrows show. It's involved in brain development. It's involved in peripheral nervous system development. It's involved in GI and immune function as well. And so if you have a variant that's putting your brain at risk during development, it's logical to think that it may also be putting your gastrointestinal system at risk during development as well, or even how it can repair itself over time. So we looked in the AGREE sample. There were 214 families that had GI medical data in the database. That's why these banks are so valuable. And we see that about 43% of the children with ASD have co-occurring gastrointestinal disorder. 26% of the parents, and about 9 or 9% of the non-ASD siblings. Dan insisted on putting all those zeros in. I wanted an exponential, but no, let's put all the zeros in. So this is a significant overrepresentation of GI disorder in children with autism in this sample. And this is work that we collaborated with Margaret Bauman and Tim Bowie up at MGH and Jim Perrin to try to understand and try to bring some validity to this idea that it is overrepresented. And then we brought the genetics to it, and we looked to see whether there was a difference in the inheritance of this allele in families where there's co-occurring GI and autism spectrum disorder versus families where there's only a child with ASD but no co-occurring GI. And that's what you see here. The red box indicates that there's overrepresentation of transmission from parent to child when you have a child with autism spectrum disorder and gastrointestinal disorder. So there's overrepresentation. We're stratifying the population down now because we think that what this allele, this common variant is marking is an underlying complex biology in which you have changes in brain development and changes in gastrointestinal function traveling together. And in those individuals where they don't travel together, this may be a very minor risk factor. In those individuals where it is traveling together, it may be a major risk factor that together with environmental factors generates the complex disorder. I'm going to skip that. So what are the implications of this variant? I said that this is important. It's one thing to identify the gene, it's another to say, okay, is that variant changing anything about the biology of the system? I got a yellow light. Okay. And I always go through yellow lights. It's a good thing my daughter's heard me talk before because she gets embarrassed when I tell jokes or anything like that. So Dan did some experiments to show that if you have the C at that site, which is 20 base pairs up from where the gene is first transcribed, it actually changes the level of gene expression in experimental systems about twofold. So if you have the C compared to the G, you make twofold less of this gene, putting you at a point where your ability to signal through that system may be compromised. It may not cause a disorder automatically, but it may be compromised enough that when other factors come along, you hit threshold. And I'll show you what I mean in a minute. This is the expression of the gene in mid-gestation human. This is from the University of Maryland collection, a collaboration we have with Dan Geschwin, and it's pretty remarkable. This is the expression of the MET gene at mid-gestation in the human brain, and it basically at this age it's almost exclusively in the cortex, it's only in the temporal lobe. The areas where language develops, the areas where the relationship between frontal regions control some of the phenotypic features that Peter talked about. This is important, not just for this gene, but for other genes, because vulnerable circuits, in part, are going to be defined by where the genes are expressed. So it's not trivial to understand where they're expressed, and we know there's not a one-to-one correspondence between the mouse and the human. So doing this kind of work ends up being important, and I couldn't have drawn a better picture if I had a Sharpie myself. We also know then, by analyzing temporal lobe from postmortem material, that there's about a twofold reduction in the levels of this protein that's expressed. So we've gone from the genetic variant to showing that the variant causes a reduction in expression, and then we've gone to the patient population in postmortem material, and there's not a lot of brain material in the brain bank, so these are very difficult and special experiments to do that we and others are trying to accomplish, and there's about a twofold reduction in expression. I'm going to skip that because I want to get to this. So what do we think is happening? Our model that we're working on, because it's hard, we have to take a reductionist view when we're doing neurobiology. It's hard to explain the entire disorder. We think that MET is highlighting a problem in the signaling pathway of Pi3 kinase. There are other genes that are clearly involved. This data not just comes from our work, but from other work that's shown that there are private or rare mutations in some parts of the signaling system. We know that what this does is it's causing, we believe, susceptibility to environmental and maternal factors that together with these risks may be leading to the dysfunction of the central nervous system development, GI, immune, and others, which eventually leads to disorder. I'll end with one example. This is an experiment that Darrell Hood and Dan Campbell in the lab did, and Darrell will be talking this afternoon, in which they exposed cells that had either the G or the C variant and looked at gene expression. The light blue bars are the G allele and the dark blue bars are the C allele. You can see there's half the levels of expression, right? But what you notice is that this chemical that was used, benzoate pyrene, impacts gene expression, whether you have the G or the C. We were kind of disappointed by that. Well, if you have the C, maybe you'd be more susceptible to benzoate pyrene or other toxins in the environment. But there's another way of looking at this data. Let's say the hypothetical threshold for the disorder is where that dotted line is. If you have the C allele, you're closer to that threshold. You don't have the disorder, but you're closer to the threshold. And now, when an environmental agent comes along, it pushes the system down below threshold, and just like a number of other characteristics of diseases that we know about, that's what's critical in terms of expression. So again, I want to thank my collaborators, Dan Campbell, whose work I highlighted in this talk, and I'm looking forward to a very exciting day. Thank you very much.